agonist, partial agonist, inverse agonist. A lot of these terms maybe you've heard before, but we really want to drill down what they mean and then going over their mechanisms. And then, of course, some terminology. We'll go over side effects and different individualized drug response, another little topic we'll cover. So, and also the dose response curve, potency, amplification, a lot of traits that have to do with the mechanisms of drugs and how they work. So first, we'll cover the basics of what is a drug. So a pharmaceutical drug, by definition, a medicine or medication, a compound, a molecular substance, whether it be a small molecule, large molecule, a piece of DNA, piece of RNA like a vaccine, different gene therapy, antibodies like biologics, anything, any molecule, that nanoparticle that is used to treat, cure, prevent, or diagnose a disease, to promote health, right? That's what a drug is. Very broad category. And now the World Health Organization, who defines it as a substance or a product that is used or intended to be used to modify or explore the physiological systems or pathological states for the benefit of the recipient. I like that definition a lot more. It gets into the physiological systems. So a drug, what we're doing with a drug is we are modifying biological responses. We are modifying signal transduction. We are modifying gene expression. We are modifying some kind of physiological process to benefit the recipient, to maintain homeostasis, to promote well-being. If we're hypoglycemic, we're going to take a drug to increase our glucose. So it's a maintain that homeostasis, those levels. And now each drug, when it becomes ready for consumption. So if you take a pill with different, like a caffeine, right? I mean, don't take caffeine pills. It's not generally good for you. But I showed you the structure of caffeine last class, where, let's see if I can get my pen up. There we go. So caffeine is a bicyclic structure like that, and it's got a couple of these on it. Got carbonyl groups, nitrogen. It's aromatic. That's too many bonds, but it's aromatic. There. Yeah. So that's basically caffeine with a couple nitrogen. Yeah. Whatever. So if you take a pill that has caffeine, and it's a caffeine, marketed as a caffeine pill, the active ingredient is caffeine. Most of the pill is not caffeine. A large majority of the pill, and it depends on which drug in terms of percentages, is most of it is the inactive ingredients. So every medication has inactive ingredients. So these inactive ingredients, they are biologically safe. They have to be biologically safe, or else they cannot be inactive ingredients. And they help the active ingredient get to the target site of the drug. That's one of their uses, but that's something that we keep in mind when we're making the drug. If the inactive ingredient is something that would bind to the active ingredient, then the pill wouldn't be a good form of a drug delivery. So that's the inactive ingredient. Now, who can think of maybe a couple of other reasons, maybe brainstorm, a couple of other reasons of why we would have an inactive ingredient? So I mentioned that it's important for, maybe it facilitates the drug getting to its target site. So you could think and answer this. I want to hear some thoughts. How does this happen? Or other reasons why we have an inactive ingredient, other purposes. Inactive means placebo. Not really. So a placebo is something that is given to a control group to mitigate any psychological effects of taking a drug in the event of a clinical trial. So it's and the inactive ingredients are like salt or something, like a polyethylene glycol, or different types of salts or buffers that are in the drug pill that is taken in order to have some kind of, or in order to bring the drug or bring the drug to where it's supposed to go. Carrier,
so does it act as a stabilizer to help the body absorb the right active ingredient?
Exactly. Good. And also, a bell carrier. Yes, that's exactly it, Daniel. You're both right. So it could be a carrier, and it can help the drug and protect the drug, and make it and help it get to it, facilitate its action in several ways.
Professor.
Yeah. Yes.
Sorry, I didn't realize you were listening to me off.
Okay. I'm sorry. Yeah.
Yeah, I was just going to say, also maybe logistically, for instance, I know in certain biologics they have to freeze it, and there has to be a specific buffer that you can still freeze the drug, but it doesn't damage the drug, so they suspend it in a certain buffer depending on what kind of medication you're working on. So, and that, I guess that could be classified as part of the inactive ingredient list.
Yeah, that's perfect. Yeah. So getting into that's another facet of it. I'll talk about that in a minute. That's awesome, Ben. So what Daniel and Abel said are kind of on the same track with carrier. So you can have drugs that are very frequently metabolized by in the gut biome, or through first pass or second pass metabolism in the liver, or by the cytochrome P450 system, or by other enzymatic reactions in the cell. Especially when you're dealing with antibiotics that have specific enzymes in bacteria designed to cleave them and break them down. So these inactive ingredients help to protect and actually add protecting groups to the drug, to the active ingredient. So and let it bypass the first pass metabolism or second pass metabolism in order to get to the systemic circulation to allow it to go through its function. So that is by direct protection of physically having other chemical groups bound, other functional groups bound to the drug to protect it. And also, like you said, Ben, and also Daniel, the carriers or buffers. So these buffers are not only important in the body to help the drug, or if it's an antibody, or even usually antibodies, or a peptide. Antibodies or peptides are different types of protein mimetics, or different kinds of protein-based, amino acid-based drugs, or even RNA or DNA types of drugs. They're going to have their structure, their three-dimensional structure, and that integrity of the three-dimensional structure is what gives them their function and gives them their binding capacity for whichever biological target that they are going to bind to. If that three-dimensional structure, that tertiary or quaternary structure, gets denatured, even if the bonds don't break or cleave, even if that gets denatured, the drug is not going to go through its function, because antibodies are large proteins. They have very specific binding sites that are all based on amino acid residues that can easily be metabolized and also can easily be denatured, which is why the storage conditions super important. That's why buffers are important. Also, when you consume it, the buffer system that the pill is taken in better defend against the gut biome. And once it breaks down, or once the pill breaks down, the antibody or peptide drug should be able to pass through get to the systemic circulation in order to have a biological outcome. But generally, peptide and biologics and antibody drugs are usually injected, because it's very difficult. Really, an antibody is going to be metabolized when it's taken by oral. So it is very difficult to get around that. But yes, and also to Ben's point, when we get to manufacturing. So when you're developing and manufacturing these drugs on a higher scale, they may easily denature, or they may not be active in certain buffer conditions. They might need 20% ethylene glycol in order to maintain their activity. So in these large scale, these big chemical plants where these drugs are mass produced, maybe they need that in the buffer. Maybe they need other things, some reducing agents, DTT or TSEP or BME, other things like that when we're making a lot of these drugs. So all of that is why we need inactive ingredients to protect the drug, and also to help the mass production of the drug, and also to in all get the drug where it needs to go. Sticky, normally surfactant. Yep, surfactants are added to the final drug formula to ensure dose delivery presents the AAV from sticking to the syringe. Yeah, exactly. So that's another example. Great job, Lakin. Great job. So yeah, yeah, awesome. Okay, cool. Any questions on that? On active and inactive. Shelf life. Yeah. So Abel, great point. So shelf life, we also need to keep that in the manufacturing process. We also need to keep that in mind. That if we have a drug that only has a couple weeks shelf life before it loses activity, we need to implement buffers and implement maybe surfactants or other biologically inactive substances, but biologically safe but inactive substances that can keep the drug active. So these are all great thoughts. And this is one of the homework questions, actually, on why we need inactive ingredients. So great that we had this conversation. All right, so Advil, we'll use an example. So ibuprofen, here's the structure right here. So in any pill of Advil, it's not 100% of this structure. So what Advil does is a non-steroidal anti-inflammatory, for those of you who don't know. So most of the strongest anti-inflammatories are going to be our corticosteroids. So hydrocortisone and hydrocortisone is the main one. Also, what am I thinking of? Progesterone and prednisone. So those are main of the steroidal ones, the corticosteroidal ones that are glucocorticoids, and they follow that pathway. But the non-steroidal anti-inflammatories like Tylenol and Advil, they are COX-1 and 2 inhibitors. They're proteins that, or enzymes that regulate the production of prostaglandins, which are needed for inflammation. So if we inhibit COX-1 and 2, we will downregulate immune responses. And these immune responses are produce inflammatory prostaglandins. Also, we have leukotrienes and things like that. So result from taking Advil, you have reduced inflammation and also less pain. Our active ingredient is the ibuprofen up on the top right. Inactive ingredients, we have a lot of biologically inactive buffers, surfactants, waxes, things like that. So we have silicon dioxide, carnauba wax. Never even heard of that, honestly. Different sugars. There's some salts in there, lactose, cellulose, polyethylene glycol, and there's actually a bunch more, depending on which manufacturer of the ibuprofen it is. So, I mean, we kind of answered why we need them. So transport, and in the notes section here, I wrote down a little note for you guys for the homework. So transport, protection. Also, something we didn't mention, smell and taste. That matters. What if ibuprofen in a pure form smells like crap? Will patients want to take that, even if compared to Tylenol or another drug? Even if Advil works amazing, I would rather take Tylenol if it smelled like nothing compared to Advil if it smelled really bad. So you need to maybe add other waxes or other non-biologically active substances to make the smell and taste better. So that's another thing to keep in mind. That's all a part of drug discovery. Okay, so are any more questions before we move on to types of drugs? Okay, cool. So this is a big chart. We're going to cover it in depth, and then we're going to go into each type of drug individually. Using an example, we'll use the opioid pathway. I'll teach you about that. And then we'll go into using it as an example for each of these types of drugs. So, drugs in total are either antagonists or agonists. Oh, sorry, about mordants. I don't think I'm familiar with mordants.
Yeah, just like the dyes.
Oh, like dyes? Yeah. Let me look it up. Yeah, because so in drug, mordants and dyes. Okay. Yeah, definitely. Because when we're developing pills, certain colors appeal for certain uses. Now, I'm not in that field, in the manufacturing field, drug manufacturing, but I know that's a really important factor. That's a really good point there. That color plays a big role. If you had a pill that's like brown, right, compared to a pill that's blue or red or purple, right, you're gonna want to take a more brightly colored pill on average. On the consumer, on average, will take something that maybe looks more appealing. So
yeah, I know they used to use that for the manufacturer of kids medications before. So I don't know if they still use that. That's why I'm asking.
I'm sure they do. I'm looking up right now to prepare fabric connection. I'm looking real quick. I'm curious too.
Okay, no problem. Thank you.
No problem. Yeah. Some of them are not anymore because, according to what I'm seeing, a lot of them consist of heavy metals. Some are a small concentration of heavy metals, but there are biodyes and biomordants that are used in drug processing.
Okay,
So,
thank you.
definitely. Yeah. Very good point. Okay, good. All good stuff. Okay, so now looking at the different types of drugs, you have two classes that can be broken down into several classes, right? There is a drug that is, we're talking about traditional drugs that bind to an enzyme, a protein, a receptor that mediates a biological response. You can have two outcomes. The first outcome is the biological response could be amplified, or the endogenous response or receptor could be activated. These are called agonists. If you have a biological response or receptor that's deactivated, these are called antagonists, right? Agonists that activate, antagonists that inhibit. But it can be broken down to several classes. Now let's follow, going from the left to the right, we'll follow each part of this kind of flow diagram. So drugs that are antagonists, and I crossed this off for a reason, because I like this chart, but one of the parts was wrong, and I'll explain why, where this X is. So antagonists, we'll go from the left. So antagonists bind reversibly to agonist receptor sites. So what that means, these drugs are going to bind competitively right where the endogenous ligand, and a ligand is something that binds to an active site of a receptor, or the endogenous ligand, it will bind competitively to replace this endogenous ligand, leading to a blockage. So the endogenous ligand that naturally binds to, let's say, the opioid receptor, in that case it may be endorphins, or if you're taking drugs, illegal, or if you're taking prescribed opiates, or if someone's doing recreational opiates, it will bind to the opioid receptor. But you can take a competitive inhibitor or competitive antagonist like Narcan, for example, that competitively binds and blocks to the opioid receptor, not allowing any opioids from binding and going through their mechanism and leading to hallucinations, obviously decrease in pain and feeling of euphoria, and even respiratory depression when you overdose. So antagonists competitively block an active site. So it says competitive antagonists block effect of agonist to activate endogenous transduction mechanism. And then it says here, effects can be surmounted by high agonist concentration. So if you have a competitive antagonist, if you keep taking that illegal drug, that illegal opiate, there will be a concentration point to where the drug will overtake and recompete and bind back to the opioid receptor, even if you take Narcan for an opioid and specifically. So if those of you who watch a lot of these, the police show, the EMT shows, a lot of the drug shows, there's been times where they have to give, I've seen it on TV, where they have to give a patient several doses of Narcan because they are so OD'd that they need to reach an effective concentration of the competitive antagonist in order to competitively inhibit and kick off all of the opioids from the opioid receptors in this person's cells. So it's a competition. It's an active site competition between an antagonist and an agonist. So what we have here also, we have the effects will be surmounted by high agonist concentrations produce a parallel shift of the agonist dose response curve to the right. So we'll learn about that later. We'll learn about ED50 later. So that's the effective enzyme, the effective concentration that leads to 50% of the enzyme activity. So any questions on competitive antagonists for now? And again, we'll cover dose response curves soon. No? Okay. Also an antagonist. Those are competitive antagonists. There are also allosteric antagonists. So allosteric meaning we're talking about binding to a different or a non-active site of the enzyme or receptor. When this happens, this may cause a conformational change in that receptor, leading to a decreased response and a decreased binding capacity of the endogenous ligand. And this is called, we call this non-competitive antagonism. And it prevents the agonist from activating because it changes the shape of the active site. So let's say do I have my pen here? Yes. So let's say before the active site, looks like this, and the drug is like that, it fits in. Let's say you take an allosteric antagonist that binds here, but it brings this amino acid chain over, and it causes a few conformational changes, and it changes the shape of the active site, which inhibits the binding of the endogenous ligand. So this is a way of what we call non-competitive antagonism. It prevents the agonist from activating and activating the transduction mechanism, resulting in reduction of the maximal effect, producing a reduction in the height of the agonist dose response. So the main difference here is that although the agonist is not binding, it will bind at a high enough concentration. But and keep in mind, we have a population of these proteins, of these enzymes. This is one of them. In, let's say, 10,000, you may have a couple hundred that aren't changed, that the non-competitive antagonist did not bind or didn't cause that much of an effect. Meaning there will still be some kind of basal level of binding by the agonist, but the total overall response physiologically will not be as great. And we'll see this again. Once we get to the dose response curves, we'll see this in action. But what I have here as an X is it says an antagonist binds irreversibly to an agonist receptor site. That is not non-competitive. That is competitive. So here it says it binds reversibly, we covered. It can also bind irreversibly. But irreversible. So if you have a drug that binds irreversibly to an active site, it is most certainly a competitive antagonist. It is not non-competitive because it competes directly. So I don't know who wrote this chart, but that part's not right. But the rest of the chart's pretty good. So any questions on antagonists? And of course, we'll cover this again. Next couple slides will be a little bit more detail on different types and examples. But any questions for right now on antagonists? No? Okay, cool. Next, agonists. So agonists are the type of drug that leads to an activation of the endogenous signal transduction pathway. So an agonist will bind reversibly to the same site as the endogenous agonist. So let's say opioid, like let's say codeine, right? Codeine and endorphins. So endorphins are natural opioids that when we work out, when we exercise, when something good happens in our life, of course, dopamine is the main neurotransmitter, but we also release endorphins that give us this kind of euphoric feeling, very low euphoric feeling compared to codeine or or another type, like heroin, but it definitely it's a part. Well, endorphins is technically a partial agonist. Codeine is a full agonist. So we'll talk about the difference there in a minute. But they both bind to the same receptor and bind to the same agonist site, and they cause the transduction mechanism and produce a biological response similar to the endogenous ligand. And we have our sigmoidal dose response curve, and we see our ED50 and our maximum response. And I'll show you that soon. So yeah. So any questions on agonists for now?
Yeah, I posted mine on the chat.
Oh.
What?
Morphine. Yes, morphine. So codeine and morphine are very similar. So codeine is actually the same as methylmorphine. So I think we did cover this last class. So codeine gets metabolized to morphine. So these are very, very similar. So they work the same exact way. But so codeine can bind to the opioid receptor, but morphine is the active drug that binds to the opioid receptor more. So morphine is not taken orally. It's taken by a morphine drip usually, or injection. Codeine is taken orally because it's a prodrug. Like we talked about this last week, so great that you mentioned this. It's a prodrug, which means that once it's metabolized, it is now in its active form as morphine, and then it goes into systemic circulation and causes its biological response, pain relief or extreme pain relief, and also euphoric and also, unfortunately, addictive qualities. Okay, great job. All right, so I picked opioids to talk about today, to use this as examples to teach a lot of the different types of agonists, antagonists. So where's your morphine? It is oxymorphone, similar, but hydromorphone. Where's morphine? Well, it should be there. But anyway, so the way I'm kind of gonna go about teaching different concepts throughout the course is taking an established drug system and signal transduction system and applying the concepts we learned to that system. So not only you know the terms, but you also learn a couple of drugs along the way, or a couple of systems and mechanisms along the way, and you can apply those terms and concepts to that system. And I'm going to use ones that are biologically relevant or societally relevant. We'll talk about COVID a lot in this class. We'll talk about cancer. We'll talk about opioids, of course. We'll talk about painkillers, antidepressants. What else? Caffeine, even a coffee. Cannabinoids, so like weed. We'll talk about the biological mechanisms for that, CBD oil, stuff like that. So a lot of these are societally relevant now in today's research. Alzheimer's drugs, that's a huge field right now in groundbreaking pharmaceuticals. So these are going to be all topics that I use to lay the land for the concepts. So we'll learn about
Dr. DePaolo, I have one quick question. Are we going to
sure.
go over like the negative side effects of some of these drugs and why it happens?
Yeah, we can.
Okay.
Yeah. So yes, we'll do that for opioids right now. So the main negative side effect is addiction. Right here. I'll tell you why that happens in a second. So the way opioids work. So on your neurons, we have pain receptors throughout our body, as you can see from the top left. So we have the pain receptors are all in our tissues, are in our extremities. Basically, we have an ascending and descending pathways for pain. The pain pathway goes to our spinal cord, goes to our brain, and then it comes back down. This is all mediated by neurons. And we're not going to go into the super details of the neurons, but we're talking on the molecular level. So this is all mediated by neurons. You have the synaptic cleft, which is the way two neurons communicate with each other by neurotransmitters. And also by action potential. So one neuron generates an action potential, which then causes the release of neurotransmitters that reach the second neuron, which then can which then can depolarize to allow a second action potential to go down that axon and lead to a cellular response, or lead to a feeling of pain, right? So the job of opioids, I know this is mediated by in the brain and in the central nervous system, the spinal cord and the brain. This is mediated by two really important neurotransmitters. The first one's called glutamate. Glutamate is the most important central nervous system excitatory neurotransmitter. So anything you think of exciting the neurons, whether that be sympathetic nervous system involvement, so fight or flight, or extreme pain, or really any pain, we are releasing glutamate because those neurons are firing and they are sending pain responses throughout our body and to our brain and telling us to get your finger away from the hot flame. Also, substance P, which is another one that's similar to glutamate, but glutamate's the main one. CGRP, that's there too, but not as important as glutamate and substance P. Because they have, glutamate has several receptors that bind just glutamate. And then here's two of them right here, the NMDA receptor and the AMP-A receptor. Also, there's the kappa, the Ka receptor. That's another one. But glutamate's super important. And then you have substance P has the NK1 receptor, and then you have the CGRP receptor, which doesn't do that much, honestly. But the main one is glutamate, substance P. Okay. So for pain to transmit, we want the influx of calcium to depolarize the neuron. Once you depolarize the neuron, you have this synaptic vesicle. The synaptic vesicle houses all of the neurotransmitters. Once the ionic gradient is enough, and the electrical potential is enough for the neuron to fire or to propagate an action potential, the synaptic vesicle unloads its neurotransmitters into the synaptic cleft. So this is called the presynaptic neuron. I'll put pre, and then this is post. The presynaptic neuron has the neurotransmitters that go through the synaptic cleft, binding to receptors on the surface of the postsynaptic neuron, which are here. All these different, this plethora of receptors. There are a bunch of different receptors. This then causes the depolarization of the postsynaptic neuron, causing a propagation of that nerve signal. Okay, that's how the endogenous pathway works, basically. Are there any questions on that right now? Because now I'm going to bring in opioids, and then it'll make sense.
Could you reiterate what you just said?
Sure. So, in essence, the presynaptic neuron, once it reaches a high enough electrical potential, it shoots out these neurotransmitters, glutamate and substance P and CGRP, into the synaptic cleft, which bind to different types of receptors on the postsynaptic neuron that allow the postsynaptic neuron to generate an action potential, leading to neuron firing. That's how the signal propagates.
A quick question.
Yes.
How many of these terms do we need to understand, or at least know off the top of our heads, like the synaptic cleft and things like that?
Good question. Everything. No, I'm kidding. Not everything. So I would say just general concepts. So for an exam, we don't need to know any of this, honestly. I'm not going to ask you questions about this specific pathway on exams. I may give you pathway diagrams and ask you, what type of drugs would you develop to stop this? And by looking at the pathway, you could be able to say, oh, an NK inhibitor can block that, or something like that. But in terms of memorizing any of this, I don't require that. The main bulk of this material is like what is an agonist, what's an antagonist, interpreting a dose response curve. Those things I would expect to be memorized. So this is just setting the stage for an example, also to just teach you guys more.
I got a quick question.
Sure.
So what happened when irreversible agonist binding with receptors? Like, yeah, would you produce a continuously stimulus, or just waiting for the receptor be digested?
Right. So if you have a reversible, you said reversible antagonist?
Yeah, irreversible and
Or irreversible.
yeah.
So, right. So good thing. Good question. Great question. So irreversible antagonists, you're exactly right. They are covalently bound. They're what we call covalent inhibitors. Covalent inhibitors are not too prevalent, or we try to avoid them in drug discovery because of that fact that they don't come off. So we have to wait for receptor internalization, maybe by beta arrestin or by lysosomal or proteasomal degradation. And then the receptors will be re-expressed. That usually takes about a week, maybe 2 weeks, depending on the type of receptor. So we try not to aim for that, but if you have irreversible drugs, that's what's going to happen. Really good question. Okay. So now let's take a look at the receptors. All right, so now we understand that basically these neurotransmitters are released into the cleft, bind to the receptors, activate postsynaptic neuron, and cause the pain signal propagation. Enter the mu-opioid receptor. They are located on both the pre- and postsynaptic neuron. These mu-opioid receptors act to dull pain by binding endorphins and dynorphins and ankylophen's. Yes. So endorphins is the main one, though. These are the endogenous neurotransmitter or endogenous opioids. Once something binds to the active site of the opioid receptor, and this is a GPCR receptor, if you recognize that shape, but we'll learn more about that later on, about different types of receptors. But basically, it activates a downstream signal cascade, which leads to the closing of calcium channels. If you close calcium channels, what will happen? The neuron will not have enough potential generated to release glutamate and substance P from the synaptic vesicle, meaning this big red X that the synaptic vesicle will not open up into the synaptic cleft to release the neurotransmitters will take place. And then that will stop the binding of those neurotransmitters to other receptors that allow calcium to come in, that allow basically calcium and sodium to come in order to generate that action potential again. It will stop completely. So this means that the pain signal will decrease. Also, if you have opioid receptors on the postsynaptic neuron, they will open up potassium channels and allow potassium to efflux and leave the cell. That will hyperpolarize the cell, leading it to have a decreased likelihood of generating an action potential. So this will decrease and ultimately stop the pain response. So what's the point? Why are opioids powerful and may lead to addiction if we already have endorphins and these kind of substances in our body? Well, endorphins are what we call partial agonists. So the body, the way evolution worked with creating these biomolecules for our body is that they activate the mu-opioid receptor and lead to a decreased pain response, but their response is limited. It gives a partial response, maybe 25 to 30% of the euphoric feeling we feel if we take fentanyl or any of these other drugs, or any of these codeine or drugs like that, or opioids. So they're partial agonists. If we take, and also they're specific, nothing is they're specific. They're specific to this mu-opioid receptor. You notice there's a mu here. There's also a kappa and a sigma opioid receptor. the activation of the kappa and the sigma receptors, they are the ones that lead to hallucinations, more of a euphoric feeling than the mu receptor. The mu receptor also has that property, but not as much as the kappa and the sigma. Or sorry, mu. No, that is mu. Mu, kappa, and sigma. Yes, those are the three. Okay. So the opioids that are given by drug, by codeine or take fentanyl or whatever, those bind non-specifically to these receptors, the mu opioid, kappa opioid, and sigma opioid receptors, not only leading to, and also with greater efficacy, not only leading to a tighter binding and a more non-specific binding to these three receptors, but also giving a higher response in activation, leading to 100% euphoria, maybe hallucinations. You feel numb, you feel whatever. And that may lead to physiological addiction, which means what happens is if you keep taking those kind of opioids, the receptor will internalize, because your body will be like, your neurons will be like, okay, we have too many opioid receptors present, we're getting too much of a response, let's internalize some of the receptors in order to, actually no, that's not what happens here. What will happen is we don't have enough receptors to bind all the, well, either one could happen, but in the opioids case, you don't have enough receptors and to bind all the opioids, so your body overexpresses the opioid receptor, then you need more opioids to satisfy and bind to all those receptors. That's what we call drug tolerance, and that leads to addiction. Because what happens is once you're finished, and once you're taken off the opioids, you now have all of those opioid receptors, and you're hypersensitive to the effects of opioids. And also that's physiological addiction. There's also psychological addiction, that you are maybe addicted to the feeling of the feeling you get. And then this happens with a lot of, unfortunately, war veterans who are on morphine for a while. You get addicted, or people who've had, prescription opioids addiction is an epidemic in the country, unfortunately. And we see this a lot happening with teenagers even. Really, I've seen, or I've not seen personally, but I've heard stories of well-off executives or people who have had a great life that fall into drug addiction because they hurt their hip, and they were prescribed opioids and they get hooked on it. So, and then they ruin their lives from that. So it is a very dangerous drug class. And what we can do is we've developed different types of drugs that can help our withdrawal from it, and also to block the opioid receptor competitively, which I'll talk about those in the next slide. So right now, any questions on this whole pathway? Or any curious questions that you may have? Good question, Vanessa. If these drugs are so addictive, how come they're prescribed? Now, so it's being tougher to prescribe. They're prescribed very sparingly, but they're usually prescribed for a short period of time. And also, it's for illnesses that have severe pain associated with them, or surgeries that have severe pain associated with them. And there aren't any non-addictive painkillers that are stronger or as strong as opioids. That is one of the main drug fields and drug discovery projects that needs to be undertaken by large companies. Or we should, we should do? We should all make a company. We should make a pharmaceutical company together, and we should tackle this opioid epidemic. Because look, if you have a gunshot wound, Advil is not cutting it. Corticosteroids are not cutting it. You need something like morphine. Otherwise, you're in excruciating pain. That's why they're prescribed. But they can be very easily abused. So I got prescribed codeine I had a tonsillectomy a couple years back. And I took one because I think there was one night where it hurt really bad and I just wanted to go to sleep. And it's scary, but you feel like, whoa, like my whole body's like coming numb, and like, yeah, it kind of feels good. But I made sure to be very careful with how I took this drug, and I endured the pain from that surgery, and just because I was afraid of taking this drug because I know the harm that it could have on people. on even people that didn't know they could be addicted to anything. So it is scary, but it is prescribed for people with severe pain, and maybe it should be prescribed less often. And I would agree with that. With if you said that, I would agree. But it also depends on, I mean, how much pain the person's going through and how much they're willing to tolerate. So if they can create a drug that is non-addictive and is not an opioid that can lead to the same type of dulling of the pain, that would be very promising. Okay, so let's take a look at some alternatives and some things that or not alternatives, but things that can be used to mitigate this addiction situation. All right, so there's a lot going on in this slide. First is what is on the right. This is called a dose response curve. On the y-axis, we have the response of the drug. The x-axis, we have the log of the dose, so log of the concentration of the dose. And we have these letters. We have RA/D and RI. RA means activated receptor. And then RI means inactive. And then DA, this means agonist. D-ant means antagonist. I'll just put that there. DPA means partial agonist. And then DI, inverse agonist. So this part, I would suggest knowing what the concept of what this curve means. So let's go one by one. First, we'll talk about agonists. So full agonists, the top curve, the top red curve. So a conventional full agonist drugs have a much higher affinity for the RA conformation, and mass action thus favors the formation of the RA-D complex with a much higher observed effect. Basically, an agonist is going to bind to a receptor, an active receptor, or really, I don't like that they use RA plus RI, so consider all the receptors together. And once the agonist binds, it is now an active receptor. So, I mean, the way it works is, the reason why this response is not at 0 is because this is something that they won't tell you, or something that you don't really think about, is that when you have a pool of receptors in a cell, there is always an amount of basal activity, meaning there is some response, maybe 5% response, but there is some response, and there is a population of receptors, some of them activated, some of them inactivated. That's what they mean here. But once you add an agonist, all of those receptors now become activated because they're bound to the agonist. Does that concept make sense? Are there any questions on that concept? And the opposite happens with inverse agonists.
Yeah, Professor, when you say base level, are you speaking biological or base level from the drug?
Biological, not even introducing the drug yet. So let's say the opioid receptors, without any drug, maybe they're 5% active already. That's just by the molecules, like there's natural endorphins at a low concentration that are just hanging around there that activate the receptor 5%. So it's not 0%, is what I'm trying to say, is that there's always some kind of activity with every type of receptor, but it's low until you have an agonist.
That makes sense. Thank you.
Cool. All right, so it's a full agonist. So we're seeing the maximum level of response. And this is where we take our, that's not a straight line at all, but, oh God. Whatever. So not supposed to be a straight line. So we have, those are our synthetic opioids. That's not, oh, okay. I'm really bad at the straight line thing. So synthetic opioids, they're going to create the maximum response. They're going to cause complete activation of the entire population of receptors, leading to a lot hallucinations even, and it's going to be bad for opioids. But for some drugs, you might want the full response. If you have like, if you actually, not many, well, some drugs you might. But anyway, we'll talk about that when we get there. So that's full agonist. Then let's talk about partial agonists. So a partial agonist have an intermediate affinity for both the inactive and active forms. Basically, they're going to cause activation of the receptors, but not at a high enough level that they're 100% activation. It'll cause an intermediate response. For example, endogenous opioids, so endorphins. Endorphins makes you happy, it makes you feel euphoric, but it's not an intense response like fentanyl. You won't OD from endorphins because even if you took a crap ton of endorphins, artificial endorphins, if you just injected it in yourself, you're not going to get to full operation, to full activity of your opioid receptors and that response because the drug itself leads to only a partial activation. The reason for that is the structure-activity relationship, which we'll cover more when we get into specific biologic or specific chemical binding of drugs into pockets. But in general, just that specific drug doesn't cause a full response. Another one is a buprenorphine. 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 This is a drug that's used to help mitigate withdrawal symptoms of people that have been addicted to opioids. So it's kind of like the nicotine patch version of the opioid version of a nicotine patch. So it gives you that a baseline high, a baseline euphoric feeling where your opioid receptors are being bound, and you don't feel withdrawal effects and headaches and vomiting and stuff like that. But it's not enough to give you a full opioid receptor high or opioid activity high and ruin and kind of prolong addiction, you know? So that's what partial agonists do. Any questions on full agonists or partial agonists? All right, so we can say that partial agonists are less addictive if they don't fully activate. Yes, you can say that. You can say that they're less addictive if they don't fully activate. Yeah, they have less of an effect, less of a biological response. Okay, good. All right, so next we have conventional antagonists. They have equal affinity for both the receptor forms and maintain the same level of constitutive activity. So what they do is they bind to the receptor, any of them. They bind to the receptor, and they block agonists from binding. So if agonists wanted to bind, they physically block that interaction, or they allosterically block that interaction, meaning they bind to an inactive site, which causes a change of conformation of the active site, leading to no antagonist binding. So antagonists, they stop additional activity. So an inhibitor, they inhibit activity. Think about it that way. And so example is a methadone or methadone. So methadone is a drug that binds to the NMDA receptor, which we talked about over here. So it's one of these receptors that is a calcium channel. It binds here and it blocks it blocks glutamate from binding and activating this channel, this calcium channel, so it blocks that channel. It is a antagonist to glutamate, not exactly relating to opioids, but it just by itself blocks glutamate competitively. But also that methadone is an agonist of the kappa and mu opioid receptor. So it is an opioid, but I'm talking about the one specific function it has by competitively inhibiting glutamate. That's what we mean by inhibitor, an antagonist. And it is used to limit withdrawal symptoms as well. Okay. Next, we have inverse agonists. So inverse agonists have they can bind competitively to the active site, but they lead to they're basically a stronger antagonist. But it depends what the system is. So it's only an inverse agonist if it causes the opposite biological response. So for opioids, that's difficult to think about because you can have like Narcan. Narcan binds to the opioid receptor and it causes the pain response to basically increase because you have you can't even bind endorphins anymore. So it kind of does the opposite response. Another example would be a hypertension medication, right? So if you have hypertension, meaning your heart rate is fast, right? If you take too much, or if you take a beta blocker, for example, or an ACE inhibitor, your blood pressure is going to decrease. So it'll do the opposite. It'll slow it. One speeds up blood pressure, like a beta agonist. One will slow blood pressure or lower blood pressure. So it does the opposite response. So inverse agonist is a tricky one because it has to do not only stop the activation, but do the opposite of the activation. So, yeah. Okay. Any questions on that? On these four? So we have full agonist, where I expect you to know, partial agonist, antagonist, inverse agonist. So questions I would ask is, I would give a question, like a multiple choice question of saying, here's a drug that does this, this, and this. Which one of these is it? Is it a full agonist, partial agonist, antagonist, inverse agonist? Or giving you a dose response curve and asking you to fill in the blanks. Which one is a full agonist, partial, antagonist, or inverse? So things like that, just knowing the concepts and the terms. Okay, any questions on this?
Just a quick one for the inverse. So I guess a simple way of doing the inverse agonists is basically like saying A helps stop B. So what this inverse does is it stops A from working so that B doesn't, it just continues, kind of. I guess I don't know.
Yeah, so just think about it as doing the opposite. So, I'm gonna think of another example. Because there aren't many examples of inverse agonists. So if you had cancer, I guess. Yeah. There are two types of cancer drugs. There's one that could be an antagonist, which are conventional inhibitors. A conventional inhibitor will stop the tumor from growing, right? That's an antagonist. Radiation therapy or different types of chemotherapy that are necrotic or cause tumor cells to die or go through apoptosis, that's inverse agonist because it doesn't only stop the cancer from being metastatic, but it actually leads to the apoptosis or death of the cancer cells and decreasing the tumor size. So not only stops it, but decreases what's already there. That's an example of inverse agonist. Cool.
So, Professor, what class does the ACE inhibitors?
Oh, ACE inhibitors.
Yeah.
So ACE inhibitors are for hypertension, and they are antagonists for the ACE receptor. But depending on but for the physiological response, well, you may consider them as an inverse agonist because, well, it's tough to. So I would say they're okay. Depending on how you look at it, it could be both. But I would say I would categorize it under an antagonist because the activation of the ACE receptor causes an increase in heart rate and renin-angiotensin system and blood pressure, but ACE inhibitor decreases it. So technically it's an inverse agonist, but it is directly inhibiting, and the direct inhibition causes the lowering of the heart rate or of the blood pressure. So technically it's both, but I would you can have an argument for either one. But so either agonist or inverse agonist.
Okay, thank you.
And Kyle, is it more extreme? So yeah, it's more extreme, and think about it as doing the opposite. It does the opposite of what the agonist does. Okay. Yes. No. Yeah. Exactly. So good call, Ben. Yeah. So they're determined relative to the disease and also to the receptor involved in that disease. So what does the receptor do? If activating the receptor causes a response, but deactivating the receptor causes nothing to happen, then it's an agonist. If deactivating the receptor causes the opposite to happen, then it's an inverse agonist. That's a good way to look at it. All right, so let's move on. We only went through 7 slides so far, so we can move on. So a big point of drug discovery and drugs and how the drugs work is, how do we take one molecule and it leads to a cellular response and a physiological response over the course of days? And how can one molecule involve hundreds and thousands of millions of cells, or even a few molecules activate that many cells and activate all of those mechanisms within each cell to cause a response? And the answer is drugs is an important concept, that drugs cause a signal amplification. So if you activate any signal transduction pathway in the cell, it is amplified with second messengers. So, and we'll cover this quickly, I want you to understand the concept of this, that once you bind one molecule of a extracellular growth factor or some growth factor or any kind of ligand that activates this is a GPCR, an epinephrine receptor, which is a GPCR, activates it, then it leads to the activation of the G alpha S, which then leads to the release of cyclic AMP, which is, you get 200 molecules. One molecule binds to the receptor. Now you have 200 second messengers. These 200 second messengers are going around the cell, and they're activating enzymes, protein kinase A, which then leads to a kinase cascade and an additional amplification to where glycogen is being broken down to glucose 1-phosphate for energy and being broken down to glucose for energy. So, and we have one molecule can cause 100,000 glucose molecules to be entered into systemic circulation. This is signal amplification and is very important for not only amplifying within one cell, but also creating a prolonged effect. So an example is albuterol. Albuterol is used for inhalers, and it only exists in your respiration for a few milliseconds, but those activated GPCRs last for minutes because their downstream effect is propagated over time, even though one albuterol bound to the receptor. So it's that amplification and prolonging of and sustaining that signal, that a signal transduction is what causes a larger cellular response. Okay. Next, so the next thing we're going to talk about is dose response curves. Now, there are some parameters of dose response curves, and these are pretty fun, actually. I find these a lot of fun. So this is how we test and rank drugs in any stage of development. So I make dose response curves. Those of you if you've ever worked in clinical trials, or I'm sure with gene therapy, really any drug, you have a dose response curve. So on the x-axis, you're having a log of the drug concentration, or just drug concentration. It doesn't have to be a log, but normally standardized, it's a log graph. On the y-axis, you're having some type of response. It might be enzymatic activation. It might be phosphorylation. It might be cell viability. It might be, if you're dealing with mice and lethality on mice models, in vivo models, you might be dealing with lethality or anything, really. So it really be anything, or tumor size, right? So we have several parameters. We have the potency, meaning the EC50. So that means the concentration. So I'll write this down. Concentration. My handwriting sucks, I know. Concentration of 50% enzyme or 50% response, I'll put, because just to generalize it. I'm used to enzymes, but whatever. 50% of the response. So, for example, in a cell-based assay, let's say it takes 50 micromolar of a drug to cause 50%, or well, not use 50 micromolar. Let's say it uses 2 micromolar. 2 micromolar of the drug to cause 50% of the cells to die. This means its potency is, what did I say, 1 micromolar, 2 micromolar? 2 micromolar. That means its EC50 is 2 micromolar. You grade drugs and rank drugs based on their EC50s. Also, the slope. The slope matters. When we're talking about pharmacological potency and pharmacological efficacy, and also pharmacological, and also the therapeutic possibilities, clinical potential, we care about the slope. We care about if it's the drug looks like this versus this. Now, quick question. Now, we didn't even learn about therapeutic window or therapeutic index. Which one of these, A or B, you think has a greater therapeutic window? Yeah, exactly. Kyle, Ben, right. And I'm sure a lot of you were thinking A, exactly. Because if this is the EC50 right here, right, for both drugs, they might have the same EC50. Even B might be more potent, but I'd rather take A. Obviously, I don't know which illness we're talking about, but I'd rather take A. Because if you take 2 pills instead of 1 of A, your response might be here. Which, if it's like an ACE inhibitor, that's not too bad. You might feel a little bit tired, and you may not be able to operate machinery, but you won't die, and your heartbeat won't stop. If you take 2 pills of B on accident, you might be at the maximum level of hypotension, and you might go into cardiac arrest or something, or have some kind of, or stop breathing, right? So I'd much rather take a drug with a higher therapeutic index or therapeutic window. This is really called therapeutic window. Index is something different. So the slope matters. Also, the maximum, or Emax, which is the maximum effective achievable response by that drug. And so so basically the drug release into the system is non-instant. That is true, but that is not exactly what dose response is talking about. That's saying that we're not factoring in metabolism here. And we're not factoring in the time it takes for the drug to enter the body. So we can do that, though. We do have similar plots, but this is the concentration, or response versus concentration. We do have graphs to rank drugs when we talk about concentration versus time. So if you take a drug that is orally, your concentration versus time may look like this, whereas it's more time released. And this is important for your concentration max, your Cmax. So maybe those of you who've taken other classes have heard of the term Cmax. That's where we talk about the maximum concentration. But no, great that you get it. But it's a really good point. I'll just keep finishing my rambling because this is a good point. But if you take a drug that is injected, your effective concentration of the same drug might be up here. It might go like this, and then come down like that. So the area under the curve is the same, but your Cmax for the injected drug might be really high, and this Cmax of injection might cause toxicities, and it might cause side effects that are really bad. Whereas orally getting the drug may not have those side effects, even though it's the same amount of drug. Now we'll talk about that in another class, but very interesting. Okay, so we care about the Emax. Also the threshold dose, the lowest dose at which the drug is effective, that matters too. So if we care, if this line is drawn out like that, we don't even want to take a drug that is, we don't want to take a dose that's less than this. So we want to take a dose that's within this therapeutic window range, usually one or two standard deviations above or above and below the EC50. Okay, so any questions on this before we look at the graphs? No? Okay. All right, here is a graded dose response curve. There's a bunch of them. So we have drugs A, B, and C. All right. Now, the EC50s for all of them, the way to find EC50 by looking at the curve is you find about the middle. So if you want to say this is around the middle, I'd say that's around the middle. And then you go down and say, all right, which X concentration, which log or which concentration corresponds to the middle. So let's say this is 0.1. So what you would do is take the inverse log of 0.1, and the 10 to the 0.1 is what? I'm too tired to think. 1? I don't know. But whatever it is, that's the concentration that is the EC50. So that's how you find your EC50. And let's say for curve A, it's probably around here, and it's around here. For curve C, it's around here, like that. Curve D, it's probably around here. And this is all 50% response. Let's say this is 50%. And then we have up here is our Emax, or 100% of response. So if I gave you this on a test fair game, if I gave you this plot with numbers on it, asking you to find what is the EC50 of this drug, also which one is more potent. So if we have a lower EC50, lower EC50 equals more potent. So if this concentration corresponds to 1 micromolar, this one corresponds to 100 micromolar, this one corresponds to 1 millimolar, and this corresponds to 10 millimolar, then we can directly rank which one is more potent. We can say that it takes, of drug B, it takes 1 micromolar of the drug to see 50% of the maximum effect. Or drug A takes 100 micromolar to see 50% of the maximum effect. But if we're making drugs, we're also noticing the Emax. 1 micromolar of B that's fine, but it only corresponds to only 25% activation of the total response. In some cases, you want that, like for hypertension, or for maybe an opiate. You might want that. You want it to be mild, or a different types of cardiac drugs, or antidepressants, or serotonin reuptake inhibitors, dopamine reuptake inhibitors. We definitely want not the maximum response, because the maximum response for those type of drugs are elicited by cocaine, by heroin, by methamphetamine. So we definitely don't want the maximum for psychological drugs or pain relief medications. We want something in the middle. So drug B would be great. It's high potency, and also even if you take too much of it, the maximum response is only half of the activation of the receptors, or half of the maximum potential of the receptor activation. All right. So so ranking these EC50s, the lower is better, more potent, and higher is less potent. So if for drug D, you're taking 10 millimolar of the drug, you need to have that concentration in your body or whatever it is in milligrams per liter of blood, or micrograms per milliliter of blood. However you want to determine concentration, you need to keep a high concentration of the drug in your body at all times. This is not favorable for producing drugs, because you don't want to market a drug that hey, if you want to cure your Crohn's disease, you have to take this drug every hour. That's not logical. You don't want a patient to have to deal with that, and you'll lose your market share, right? Even if it is a really good drug, it's not viable for a lot of people. unless you're on in some cases it is. If you're, let's say it's COVID and you're on or let's say you have some kind of, I don't know, say Parkinson's or something, and you're shaking and you're involuntarily shaking all the time. You take a drug every hour and it cures your Parkinson's. Yes, many patients will spend all their money on that drug because the effects are so much outweigh the inconvenience of the drug. That's another part of marketing drugs that's important, with side effects, also the inconvenience of the drug, and versus what disease is it treating. These are a lot of things that manufacturers and pharmaceutical companies have to weigh out when they're answering these big questions. So anyway, any questions on the dose response curve and how it works? No? Okay, cool. So B is the most potent, but A is pretty good too. C is less potent than that, and then D, I would not take D. Is a small therapeutic window. If I take one too many, I'm all the way up here, and also it's not that potent. So A is a good drug and B is a good drug. C is okay, but yeah, C and A are the same, but this is a shift. Okay, so let's move on. Now, potency. What is potency? We talked about that just now. Potency refers to the EC50 or the ED50. So effective concentration versus effective dose. Same thing. Effective dose more relates to clinical trials. Effective concentration, we can be in an enzymatic context. We could be in a cell-based context. We could be in mice. But effective dose could be only in mice and in people or any kind of in vivo trials. And that's the amount required the concentration required to produce 50% of that drug's effect. The potency of the drug, we also talk about the KD to the receptor. So KD is a biophysical constant, and this D actually should be capitalized. And this is the kind of research that I do, biophysical. So we're talking about what is the potency of just the molecule, just the drug to that receptor? What is the physical interaction constant? And in concentration, that relates to 50% of binding. So you can have a KD that is 1 nanomolar. That's very good. But that can also correspond to an EC50 of 100 nanomolar, and it may be an ED50 of 20 micromolar. This is not a very good drug, even though you want to be in the nanomolar range when we're talking about drug potency. We know that the molecule binding to the receptor on a biophysical level is in the nanomolar range. That's great. The enzymatic or cell-based context, we need 100 nanomolar of that drug to produce the response. So it's a two-part thing, or actually a three-part thing. We're not talking about only binding. Binding is great. Binding is the first thing you look for. But also, not only binding, but how much does that binding lead to the cellular effect or the activation or inhibition of that pathway that it's associated with, or that you're trying to intervene, right? That might require a lot more concentration, a lot more molecules. So that may lead to 100 nanomolar, which still isn't too bad. But if you go into the ED50, meaning the effective dose needed to create 50% of the physiological response, then 20 micromolar may be a lot. And usually at this level, you don't measure it micromolar, you usually measure it as micrograms per milliliter of blood or grams per liter of blood, however you want to do, or usually milligrams per liter of blood, however you want to do it like that. But basically, we're talking about the transition between physical binding, how tightly that is, versus how much that physical binding causes a cellular response, versus how much that cellular response in turn causes a physiological outcome. Those are the differences between these three. And they're all pretty important to know the differences of. So any questions on that?
Excuse me.
Yes.
So is that why or is it related to what the cost of the drugs that are in nano sizes being more expensive than the other ones that are just in solid dosage ones?
Are you referring to sizes? Is that what you said?
Yeah, sizes, yeah, the particulate sizes.
Right. So particle size isn't really associated with that, but I mean, you said which ones are more? Can you repeat the question?
Like the ones, if you want to buy like some of those medications that are in gel forms.
Yeah.
So they kind of Tylenol, you know, those of them in nano sizes, they're kind of more expensive than the ones that are in regular white salt dosage ones. So is that what is that part of the thing that is put into consideration in terms of the potency of the medication?
So that is more with gels and things like that. That is more with the manufacturing process. So for gels, manufacturing process is more expensive, and also it does lead to a higher accumulation of the drug. So because that of maybe a time release, or maybe it's the gel does a better job of protecting the drug to get into the circulation, and usually gels have a time release component to them. But that doesn't really relate to potency of the drug, because it's the same drug and it'll reach the same amount.
But the sizes are kind of not the same. That's basically, I'm talking in terms of the sizes.
Are you talking about the size of like the actual pill or size of the molecule?
The molecules, okay. The molecules that are in the pills. That's what I'm talking about.
Okay. So size of the pill is really dependent on the manufacturing process. Size of the molecule, of course, that plays a big role in the transition between KD, EC50, and ED50. Okay. Because if you have an antibody, right? Well, no. Let's say we have a caffeine. Caffeine may be super potent to the AB1 receptor, right? Or no, ABA receptor. ABA receptor. Maybe super potent to the ABA receptor. Have 1 nanomolar. If you put it into the cell, caffeine has non-specific targets that are not the ABA receptor. But in order to activate the ABA receptor, we need 100 times more of it because of its small size. A lot of it may not get in.
Okay.
Right. Yeah, these are very good questions. And also like size is very important when we're talking about the diffusion through the cell membrane. Because if you have an antibody is not really going to do well at getting through the cell membrane. That's why antibodies are not going to target intracellular proteins. Usually they're targeting extracellular receptors. So they could be just stuck into your skin or into your systemic circulation, injected, and they find receptors and they bind to the receptors in the tissue. But yeah, size is a big factor. Good.
All right, thank you.
No problem. All right, so just to finish off, potency versus efficacy. Potency, we're talking about the KD and EC50, really. And so how much binding is there of the drug to the receptor, and what type of cellular outcome does it have? Efficacy is how much does that cellular outcome relate to a physiological response. So, and how much of it causes the maximum response. So how that relates. Yes. Okay. So next we'll talk about, well, briefly, we kind of already beat the dead horse with ED50. So effective dose, right? We're talking about dose, and usually in vivo trials. And then we have another one. There's actually 2 more D50s, we'll call them. There's LD50, which is lethal dose, median lethal dose. So what concentration do 50% of the specimens die? And of course, we do this with mice, not people. That would be a really bad clinical trial. And then we have the TD50, which we may do this with people in stage 2 clinical trials or stage 1, usually stage 2 or phase 2. TD50 is toxicity. So what concentration does 50% of the sample size have a toxic effect or a side effect? So here's what these curves look like. Now we have the therapeutic effect, which is your ED50 right here on the left one, your toxic effect, the TD50, and the lethal effect, the LD50. And this is the log of the dose. So each one of these tick marks is, let's say, tenfold off of each other. Let's say this is 1, this is all, let's say it's nanomolar. 1, 10, 100, 1,000, 10,000. I would take this drug on the left. Now, why would I take this drug on the left versus the one on the right? Anybody?
Yeah, Professor, your
Yes.
lethal dose is a lot farther away than your effective dose, and it's kind of close to the toxic effect, but not as bad.
Yeah, exactly. So I don't want to take any drug that if I take too much of it, it's near the lethal dose. That's really bad. So even if I OD on this drug on the left and take 10 times, let's say this is like 5. We're supposed to take 5, let's say milligrams. We're take 5 milligrams. I take 10 milligrams. The chances of me feeling any kind of nausea, vomiting, or anything, any other kind of side effects is less than, it's around 10%, right? 10% chance. I'll live with that. If I take, that's true for most drugs we take. If we take 2 or 3 pills too many, we're gonna feel nauseous. We're gonna maybe have a headache or something like that. But if it's 10%, that's fine. But just don't take too much. But if you make a mistake, that's okay. Lethal dose is how many tenfolds away from the ED50. Let's say even from 10, even from the maximum possible dose, it's 10, 100, 1,000, or 100 really. 100-fold. You need to take 100-fold higher dose to have any potential of dying. I'll live with that. If I make a mistake if I take 1 pill, if I take 100 pills, I'm supposed to take 1. Yeah, then most of the time with most drugs, even over the counter, you might die. There's a chance of you dying. So definitely, this is a much better drug. The one on the right, on the other hand, I found this chart online. I didn't see which drug it was referring to. I really hope it wasn't referring to any drug, because whatever drug this is should not be clinically approved. You have the therapeutic range is here, and say our ED50 is here. If even at our ED50, where we're seeing a therapeutic range, 10% of the patients might have some kind of toxicity. That's not very good for most cases. Now, if we go take on the highest therapeutic end, you have a 20% chance of dying. I definitely don't want to take that drug. Can you guys think of a disease that you might want to take that drug? Cancer, good. Lakin, great. Cancer, Alzheimer's if you're in like late stages, or Ebola, or anything really, really bad. AIDS. Well, I mean, AIDS now is livable, but like if you're about to die from AIDS and this drug comes out, I'm taking it. So great job. So the point here is any drug or any illness or disease that is life-threatening, people will risk more to save their own lives. Whether that be, I mean, a lot of the chemotherapy drugs have terrible side effects, but we still prescribe them. Marburg disease, exactly. So as I'm not a clinician, but clinical doctors, they prescribe drugs that may have awful side effects, but it is a good chance, or it is a chance that person's life will be saved. So in that case, we're definitely going to prescribe those drugs and we're going to take them. So good. Okay, so we talked about therapeutic index. The definition of therapeutic index, the ratio of the TD50 to the ED50. And I'm not going to really test you on that. That's a small definition. But I would just test you on, if I gave you one of these diagrams, tell me what the ED50 is, the TD50. Would you take the drug? Which drug is more safe, A or B? If I give you a dose response curve with several drugs on it, be able to compare and contrast the different drugs for potency. So those kind of concepts will be tested. We talked about this already, the therapeutic window. Also, we talked about the clinically acceptable risk of toxicity depends on the severity of the disease. So for less critical illnesses, headaches are maybe okay, but you should not really have many toxic effects. But for treating lethal diseases such as Hodgkin's lymphoma, there are acceptable toxic effects that can be tolerated. All right, so for the last thing, I mean, we're not too far from being done, but the last thing that the last idea we're gonna cover is side effects and individualized drug response. So variation in drug responses. This is something that we only find out more about in clinical trials because we're exposing the drug to a lot of people. In mice, most of these mice are the same, or genetically, a lot of them are similar. And even with normal variation, they're going to be extremely similar with respect to different people. We have people of different, even within the same race, or even within the same, let's say, same origin of the same town in 1,000 years ago, there's a lot of genetic variability. When you factor in other races, other cultures, other epigenetic factors, whether it be climate, the type of food you eat, stuff like that, you're going to have a huge variation in drug responses. And also drug responsiveness. Just took me now to realize that. The word was responsiveness, but also drug response. So you have, we'll cover the weird one first. So you have idiosyncratic drug responses, which means a very unlikely rare side effect, which these are broadcasted sometimes in commercials for drugs to say rare side effects include, like, it's usually something extremely severe, like blood clots and death. But if you look at the stage 3 or the phase 3 clinical trials, or even the post-market clinical trial, the post-market trials with 100,000 individuals, maybe one had some kind of random idiosyncratic side effect that gave them a heart attack and they died, like a day after taking the drug. So you have to mention that when you're making a drug and bringing it to market, but you also label it as an idiosyncratic drug response. So could be some kind of hyperreactivity or hyporeactivity, and it gives an intense, either in the drug intensifies or it doesn't do anything, or it gives just off-target side effects, and it's just unpredictable. So that's one type, it's idiosyncratic. Then we'll talk about more in a second. We'll talk about more reasons for side effects in a second, or individualized drug response in a second. But it's also tolerance. So tolerance varies between individuals. And it can depend on a lot. It could depend on the prior use of that type of drug, even that drug or that type of drug, or the amount of expressed receptors for that drug. Some people have the expression levels for receptors are different. The expressed levels of enzymes that are like metabolic enzymes, like cytochrome P450 or P-glycoprotein. These kind of enzymes in protein channel, P-glycoprotein is a protein channel. These kind of proteins have a difference in expression level with individuals, leading to a different tolerance for drugs. So, like alcohol is a perfect example. Like most of us have drank alcohol before and have been around people that drink alcohol. Some people have genetically a higher tolerance because their metabolism of alcohol is faster, genetically. Not even from drinking a lot, just genetically it happens. And some people do not tolerate alcohol very well, and what we call them lightweights. And there's a genetic component to that. It's not just because they don't drink a lot. It's a genetic component. And there's also an epigenetic component, including their size, their weight, their heart rate, their metabolism rate, their exercise level, their diet. There's a lot that goes into that, and it goes into tolerance. Yeah, so Lincoln, let me see. Does ED50, TD50, ED, and LD curves account for human population variation, or are they just based on monogenetic populations like mice? Good question. Now, both. You can, well, so the 3 curves are from in vivo models, but you can generate some of those curves with clinical trials with hundreds of thousands of people, usually with an ED curve and a TD curve, and you don't really want an LD curve. But usually, yes, they are generated by monogenetic populations. To make it more variable, they do have different populations of mice. I'm not too familiar with in vivo studies, because I've never done that personally, but I know there are genetic controls in place for this. So that's where they generate those curves. Very good question. Okay. So when the responsiveness diminishes rapidly after administration of the drug, the response is said to be tachyphylaxis. So tachyphylaxis. That's when you have this can be caused by several things. So basically when you have the drug doesn't work, right? That happens in some cases. Some people don't respond to Advil and they have to take Tylenol because the mechanism is slightly different. And this could be because of the negative feedback loop. For some reason, the enzymes involved in the negative feedback loop associated with that drug binding to a protein and causing a response is hyperexpressed. Those proteins can be hyperexpressed, causing a faster negative feedback. Or the metabolism can be increased. Or there could be receptor internalization or desensitization causing the drug not to have any activity. So it's variation stuff. All right, so the last thing we'll really cover is the variation in drug responsiveness is caused by 4 factors. And these 4 factors are important. And they kind of make sense when we talk about, I don't know why I put propanol there, but I don't know why I did that. This was from last year, from my other class. Anyway, but the variation in drug response, 4 factors. 1 is the alteration, oh, I know why I put it here. The alteration in concentration of drug that reaches the receptor. So for some reason, you take the pill, or propanol. Propanol is a beta blocker. For some reason, that drug is not reaching the systemic circulation. That's because of an increased amount of expression of cytochrome P450 enzymes in the liver. Or it could be your gut biome, the pH is naturally a little bit lower, causing an increase in activity of enterokinase or some other enzymes that are pH activated, or trypsin, that break down, that cleave certain drugs. So that's getting to the receptor. Not only to the circulation, but also the receptor. There's also another step of that. So once you're through the liver and through metabolism, you now have to get to the receptor. So if you have an alteration in your blood plasma, your albumin, right? So the proteins in the blood can also bind drugs. If the drug for some reason binds to let's say your blood type, it binds to more proteins in your blood type than if you're B versus A, or A versus B. Let's say it binds to proteins in blood type B more than A. A will have a higher effective concentration that reaches the receptor. B won't. So these are a lot of things, a lot of factors. So every slide, it seems like we're learning about a new factor that goes into deciding whether a drug is good enough, going from all the way exploratory to the clinical level. And that is what makes drug discovery so difficult, is that all of these things have to be considered by hundreds of people working together. The second one, variation in concentration of the endogenous receptor ligand. What if you're taking Narcan because you had an opioid toxicity event or overdose, and your body happens to express, you're an athlete, and your body happens to express and synthesize a lot of endorphins, even though endorphins, and so let's say 10 times the amount of endorphins. The amount of Narcan required to inhibit your opioid receptors is going to be a lot higher because you have a lot of competition, not only from the opioids that you overdosed with, but with the endogenous receptor ligand, the endorphins. So that variation in the concentration has an effect. So, for example, propanol is a beta blocker, and it slows the heart rate for a normal patient, or sorry, for someone who has a high catecholamine, meaning like epinephrine, norepinephrine, meaning it lowers their heart rate for someone who's hypertensive. But for a marathon runner, someone who's well trained and their expression of catecholamines is lower because their natural resting heart rate is lower, it's not going to affect them that much because their heart rate's already lower. So your propanol is a beta receptor antagonist. It's assumed that the person is already going to have, if they're assuming that the beta receptors for the patients are going to be already competing with catecholamine binding, like a norepinephrine, that is already raising the heart rate. But if you just bind this antagonist to unoccupied receptors, nothing's going to happen. And where do you see these unoccupied receptors? In marathon runners or people who are very well trained physically, because they don't need their heart rate's very low to begin with. But so they don't have catecholamine binding. So yeah. Any questions on A or B? Because they're kind of intertwined. No? Okay. Then C, alterations in the number of functional receptors. Kind of the same thing, but we're talking about the other end of it. We're talking about expression of the receptors. So how many receptors are expressed? Some people might have a lot of receptors expressed. So let's say for caffeine, tolerance, right? When you take caffeine, the caffeine binds to the ABA receptor, and then that requires, and that leads to, adenosine binds to the ABA receptor, and it causes you to be awake. Occupation of that receptor causes you to be awake. The more receptors you have that are open, the more sleepy you'll feel. If you keep drinking caffeine, those receptors will clog up and you'll feel awake. If you stop taking caffeine, those receptors will be open and the natural pathway of you feeling sleepy will be intensified. This is what we call caffeine addiction, because then you get extremely tired, or a crash. You get extremely tired. And it takes a couple of weeks for the receptors to return back to their normal expression level. So that's what we mean by alteration in number of receptors. And then D, the changes in the components of the downstream response to the receptor. Now, with a drug, you can now have depending on the individual, you could have a difference in expression of transcription factors or kinases or histone acetyltransferases or some kind of post-transcriptional modification or post-translational modification that is different through an individual, leading to different proteins being active to a different extent. So those are the different types of variation. They're the factors that lead to variation in drug responsiveness. So any questions on these four? Because they are important to know. No? Okay. And I do have more explanation of that. So here's variation. They're all four listed. And here's the basic diagram showing drug responsiveness. In the red, you have the drug. The blue is the endogenous ligand. If you change the concentration of the drug and increase it, yes, it'll have more of an effect. So the amount that gets to the cell, caused by metabolism or gut biome or whatever, plasma protein binding, etc. Variation in concentration of endogenous receptor ligand. If the blue is increased, then the red is going to not have as much of an effect, and the tolerance or the amount of drug responsiveness will be lower. Alteration in number or functional receptors. If the blue receptors are increased, the drug will have more of a response. If the blue receptors are decreased, the drug will have less of a response because there's more competition with the blue ligands. The changes in components response distal to the receptor. Distal meaning downstream. So let's say there's a downregulation of these enzymes or a downregulation of these substrates, or that'll change the effect. So in a nutshell, that's it. Any questions on that? Okay. All right, so this is basically what I said. So alterations in concentration of drug that reaches the receptor, we went over this. Variation in concentration of the ligand, we talked about this already. So this is just the same things I've said, just for your own reading for when you're studying. We're basically done. The last thing is terminologies. Now, these are some terminologies you should be familiar with when we talk more about this during class, when we talk about maybe manufacturing and stuff like that. So you don't have to, and I'm not going to give you on an exam quick that define all these definitions, but I may give you a matching or something like that. So pharmacology, the science that studies the effect of the drugs in the body. Pharmacopoeias, the pharmacopoeia, right? All of the authorized drugs available within a country. So the arsenal, I think of it as drug arsenal. Medication, we talked about that. Prescription is the written direction of preparation and administration of a drug. Toxicity, harmful effects of the drug. So side effects that lead to toxicity, or just toxicity in general, harmful effects of a drug. Drug allergy, the immunological response. So for allergies, there may be a immune response, or there is an immune response against a drug, which causes hives or some kind of inflammation response. Maybe you can't breathe, or this is generally some kind of inflammation, histamine response. So there are allergies that is again, that's an individualized drug response associated with several drugs. Like my sister, for example, is allergic to penicillin. So that's a common illness or allergy to a drug. Drug interactions. We didn't really talk about this, but we will more in depth in another lecture. But drug interactions occur when you take multiple drugs in combinatorial therapy or just combination for different illnesses. But those drugs end up having an effect with each other and may cause a toxicity effect, or it will decrease the effectiveness of both drugs or just one of them. So an example is Clonidine and Propanol. So I mentioned Propanol already, which is why I use this example. It's a beta blocker. So Clonidine is an alpha adrenergic agonist. So it's an alpha 2 agonist. Now, we didn't really talk about the alpha and beta adrenergic receptors, but alpha 2 is inhibitory. So it inhibits let's say, epinephrine. So it leads to decreased sympathetic involvement. So decreased sympathetic nervous system. So no fight or flight. You're at a rest and digest. So Clonidine increases the inhibition, leads to decreased sympathetic. Okay. So when Clonidine, so this means that when this happens, the adrenergic receptor I mean, I have it written here. That I'm just gonna. So the adrenergic receptor. Since this is happening, you're inhibiting epinephrine synthesis also. This means that there's less epinephrine in the synaptic cleft. So epinephrine is excitatory for the sympathetic nervous system, and it leads to activation. So we have no epinephrine in the synaptic cleft. Okay, so when clonidine is withdrawn, right? So clonidine, no clonidine. Therefore, epinephrine will pool up. There'll be an influx of epinephrine in the synaptic cleft. Now, what will happen here is that this will create a really amplified sympathetic nervous system response, meaning a lot of, if we go back, a lot of, on the bottom right, we see vasoconstriction, bronchoconstriction, it would be increased heart rate, increased muscle contractions, and just a lot of excitatory mechanisms, right? Now, in order for the body to compensate for these excitatory mechanisms, you will want to have a response with the beta receptors. So the beta adrenergics, beta adrenergic receptors, you may have a negative feedback loop, or you should have a negative feedback loop with the beta adrenergic receptors. But if you're on propanol, these beta adrenergic receptors are already blocked. So you cannot propagate a negative feedback loop, in which leads to an uncontrolled sympathetic nervous system response if you combine these two drugs and then you take off clonidine. So this is not healthy, and it creates a lot of problems. So yeah, that's an example of drug interactions. There are a ton of drug interactions. You know what? I'm going to add a homework question after this. I'm going to add, yeah, okay, but I'll do that when we get to it. But anyway, drug dependence. We talked about physiological and psychological dependence. So physiological dependence or addiction is because of the biochemical changes of the body tissue and the physiological systems that cause you to need to take the drugs to function normally. Or else, like for people who are alcoholics, if you withdraw yourself from alcohol, you get headaches, nausea, vomiting, you're tired. You need alcohol to function, which is not a very good life at all. And also the psychological dependence, which not really, maybe from alcohol, but not really. Usually it's physiological with alcohol. But for caffeine, it might be psychological also. Well, there's two components to each. But for opioids or any other cocaine or more hard drugs, there is a psychological dependence. So also for like THC, there's more psychological dependence than physiological dependence. I mean, THC isn't addictive, but if you have people that are addicted to it, it's most likely because of the psychological effect that it has on them, and they see it as good as a way of escape or whatever it might be, that leads them to keep taking this drug. Okay, so here's the homework, and I do want to. I'll stop the recording first.